innovative reports, learnings, findings from this meeting. And I think we might start by just asking generally if you could begin by discussing where you think the most fruitful effort should go in terms of therapeutic strategy and actually choice of therapies.
I'm happy to start. It's a good, tough question from the start. First of all, I was just so impressed by all the lectures that we heard today and yesterday, and the number of companies in this space and the different targets. I mean, I'm left with a couple thoughts. One is that it's like a big puzzle that we're putting together to understand this biology, and the more we can focus on those models of the illness or those biomarkers that give us more confidence that what we bring into the clinic is getting to its target, having its biology, and can have some type of short-term readout, the faster this field will go, because it's very likely that many of the drugs we heard of will have efficacy and they might work well in combination, but we have to find a way to do it more efficiently because of the time needs as well, but the sheer number of different ideas of what might be going wrong in ALS.
So let me pick up on exactly what you just said, and then we can circle around to the other panel members. You alluded to doing trials more efficiently, which I think brings us to the fascinating topic that you, in fact, Merit, have put on the map, which is redesigning trials to be done in parallel in the platform motif. And my question is, can you say more about that? Could it be, just to be devil's advocate, that the problem with the motif, the platform design, is it's so short that we lose access to longevity of therapy and potentially important therapeutic information, which I think is one of the big lessons from the Tefferson study. And I worry that we may be throwing the baby out with the bathwater in some of the early rapid decisions after only 6 months in the platform trial.
Yeah, I guess I have a kind of the opposite view. I would say depends on what phase drug development you're in. I think if we're in with this number of drugs, we need an approach that screens drugs more rapidly to decide what are the best ones to bring forward. And that's where, again, getting a short-term readout measure will transform this field. Everyone gives the example of MS, that they used to have to do very long studies looking at disability. And as soon as they had an imaging biomarker, they could screen drugs in 6 months. In a way, ALS-FRS is that for us. We could get better. Maybe neurofilament will be it. Maybe there'll be something else so that we can screen drugs actually in 3 months and then bring the best ones to the longer phase trials. So that's one thing. So the Healy platform trial is not a phase 3 network, a platform trial. It's a phase 2/1. And so for most drugs, as we saw in Amylyx and Edaravone, you can get a readout in 6 months. Some other ones, like gene therapies, might take longer. So it's probably depends on what phase you're in and what the target is and what the mechanism of the drug is to decide what thing. But we used to do 18-month studies, and we heard loud and clear from our patients that they didn't want to be in a survival study, and they wanted us to come up with shorter term approaches. And so we have to keep pushing that way, I think.
Yeah, I would just amplify that. I think that's right. There isn't, there may be scenarios. I mean, I guess in retrospect, we would all say, well, gee, maybe the Tefferson trial should have been at least 9 months long. I think we didn't know that going in, but the design of the trial did ultimately show us, I think, show us benefit of the drug. It didn't hit its primary, and so that's not ideal. But what is really good is that having that open label extension showed us slowing of the drug and also gave people access to the drug, so that we actually, what we saw was that everybody in the trial had some slowing in the open label extension rather than just the people who were randomized to active drugs. So I mean, I think there certainly are scenarios where one might say, we're going to design a longer trial, but I don't know that we have to say every scenario has to be like that.
Yeah, from the patient perspective, I think having the open label extension makes a huge difference.
Yeah.
I think it entices a lot more patients into the process. There's a lot of reluctance for placebo, and yes, scientifically we need that, but that presents an opportunity for the patient to be involved in the main part of the trial, as well as gaining access to the drug, and does provide that longer timeline. And I think Amylyx, that trial showed that very, very well.
Yeah. But I
I
wonder.
might have a slightly different, oh, sorry, I'll let you finish,
No, go
Bruce.
ahead. Go ahead,
Oh,
Andrew.
okay. So I agree, and I have a slightly different way of looking at it. I think we have to be driven more and more by these biomarkers, whether these biomarkers are biological, like neurofilament, like some of the others that have been proposed, and are being looked at, chitinase, some of the immune markers that are being used in a number of programs. Somebody's actually raised the issue of troponin and P75 in the urine. These are all biofluid biomarkers, sorting out whether we can find a path forward with electrophysiology that's tolerable to the patient and has a sufficiently reduced variability. The 6 month, I totally understand the need for the 6 month. I think there are, if we're going to try to make things fit into the 6 month framework, and it's really the regulators that make us keep in the placebo arms. And unfortunately, with the tofersen trial, I think we're going to end up with placebo arms for a little bit longer. The 6 months is a good length of time. It's tolerable to most patients as long as there's an open label extension. My opinion is open label extensions have to be mandatory, and they have to be used to continue to collect data, because we've just seen how important it was. We do need our clinical outcome measures. We need to sort them out a little bit better. We need to figure out how to reduce the variability in the results that are really frustrating us, and possibly one more time expand our look beyond the ALS-FRS-R alone as the clinical outcome measure we're moving forward. Now, there's a lot of work being done, really exciting and important work on harmonizing the ALS-FRS, which is great across the pond, and here people are working together and having a unified approach. But maybe we should reconsider pulmonary, and I'm still interested in strength testing, but I know I'm one of the few. But a combination of a biomarker and a clinical measure may really help us, as long as we can keep down the variability of the clinical measure through various ways. I think that's going to help us really get somewhere in the 6 months. And I'm going to make one more plug, Bob. Every trial is valuable, whether it's positive or negative. Every trial teaches us something, and every trial produces a data set that is useful to the entire industry. So this plug is for something I have no shares in, which is called PROACT. And we've loved it, but we've sort of not pushed the latest sponsors to getting their placebo arms into PROACT. And I think we have to keep that resource live, because it is the default way we're making our statistical plans. So I think Merit and her group made an extraordinary gift to the field with PROACT. We just have to keep going. But 6 months would work with strong biomarkers and a tighter outcome measure, for sure.
Thank you. I have a question about open label extensions. Should it, out of fairness, be the case that when, and actually I might ask this of Bruce, but when people have waited 6 months to get into the OLE, should they at that point be allowed to take other drugs as well as the trial drug? Or does one want to demand that there be purity for however long the OLE extends, which is rather limiting, it seems to me. I wonder if Bruce could comment, and then others.
As a patient, I certainly like to see flexibility, and the example I can give is I'm in an EAP. I've been in it for more than 2 years. And as protocols have changed over that time, I've been able to add other protocols. Yeah, and yes, we are also measuring things in that EAP. So it's a little bit different from a typical EAP. It's almost more like a clinical trial.
Thank you.
I think it depends on the goal of your study again. So if it's really a phase 2 study and you're using it to guide to go to phase 3, then it doesn't really matter that much on OLE. I think you'll gather some other information. But if you're using it like Amylyx used it, where it's to provide other confirmatory data for a single study approval, then if you are less strict on the open label, you're not going to be able to use that data. So so it really depends on the goals of the study.
Thank you.
I think that's right. One of the challenges, though, is that, you know, for a reasonably sized phase 2 trial with an open label extension, it may become part of a submission package, even if it's not the only, you know, a single trial approval. It could be a part of that package. So it's a tricky one. I absolutely see the impetus, and you know, Bob, there was, you know, you asked that question with prejudice in a way, and I think we all appreciate that where you're coming from, and it's something we talk about a lot.
Yeah.
But every phase 2 is going to be part of a package if we're moving it into phase 3, right? Because it's getting away with one pivotal program is based on having really good phase 2 data. That's how the orphan drug sort of pathways work. So, but if a drug is approved in a country, there's a point at which we cannot not allow its use. And if an OLE starts looking like, you know, successful drugs like they did in MS, where the OLE for Gilenya lasted 8 years in some countries, you can't do that in a disease like this. So.
That's the point. I wonder, just picking up on a point that you had made, Angela, if James, you could comment a little bit on the use of these combined outcome studies, the CAFS concept, and just say a few words about that. I think A, what is the concept, and B, should we be doing more of that kind of primary outcome?
So it's a great question. So the CAFS really came out of, I mean, really the CAFS came out of the idea that in order to choose a primary, there was some risk. You could choose survival as a primary. We were doing longer trials, so you could choose survival as a primary. You could choose ALSFRS-R decline as a primary. But either way, you run the risk that you don't hit that as your primary, hit the other one as your primary, though, and then you're going to the FDA to say, well, listen, you know, we thought survival was going to be the thing that was primary. It turns out we didn't hit on survival, but we did on the ALSFRS-R. And so that was one of the main motivations, and the other one is just that especially in longer trials, there's more loss of participants, both to dropout and because of survival events, and that the methodology for sort of carrying forward ALSFRS-R or analyzing it in when there's meaningful dropout, not sort of dropout at random or loss at random, is complicated. And so that was the other problem that it was sort of meant to solve. And, you know, if we look at the CAFS, so the CAFS is one way to do this, I should note. It's one way of combining function and survival. So it stands for combined assessment of function and survival. But there are other ways that I'll talk about in 1 second. But, you know, the idea is that in certain situations, if you have an effect on both survival and function, you actually gain power. But there are scenarios where you also lose power with the CAFS analysis. And one of those scenarios is if you don't have very many survival events. And when we move to mostly 6-month trials, we reduce the number of survival events we see. And so the CAFS, the sort of benefit of the CAFS remains, but is somewhat mitigated in a shorter trial. And so I'm not sure that it's always the right endpoint. Now, the platform trial uses a different approach to this, which is a sort of a Bayesian analysis that analyzes ALSFRS-R accounting for survival. So really exactly the same goal. It just does it in a slightly different way. And I think it makes better use of the function without sort of relying too heavily on the survival. And so it provides actually that benefit as well as a few other benefits the platform trial. This all comes from actually work that David Schoenfeld did in HIV, combining survival and CD4 counts, actually. So it's sort of any kind of continuous variable and survival. And I think it leaves open the possibility that we can incorporate things like neurofilament if we think that's important, or strength testing, or whatever it is that we think are the important endpoints. I think we can find ways to analyze them together and combine endpoints.
Mhm.
And one of the things that I'm excited about for the platform trial is that we actually will have big data sets that will be able, really complete and big data sets that will be able to sort of ask some of these questions about how would these endpoints.
Excellent. Yeah, but let me bring up another issue related to trial design that has actually also come up in the chat. And that is predicated on the talks we saw today wherein 2 drugs were looked at. So obviously Amylyx comes to the fore as a combinatorial trial. And as we saw, these have been effective in other diseases like Parkinson's. So where are we in terms of thinking about combinatorial drugs?
I'm happy to start. Oh, Merit, you want to?
No, go ahead.
Okay, I mean, I'm happy to start. It's a really, I think it's a very complex question. I think back to actually the meeting that Hiroshi Mitsumoto put together, maybe, gosh, it must have been 5 years ago at Arling House, about trying to come to some guidances or guidelines for clinical trial design. And I think this was one of the sections that got the most robust and boisterous conversation. And that typically happens when nobody knows the answer, really. And I think that's probably the state of it. It does seem like a good idea. And there's no problem in doing the trials. You can do them. I think where the conversation comes is how do we do them efficiently?
Mhm.
And really our current trial designs, guys, are add-on trial designs, right? So we are not where we were 15 years ago. We are now adding on to the approved drugs. So it's a slightly different scenario. So we are doing drug combinations, but the actual combinations are based on what's approved in the particular country, rather than doing combinations. And I still believe that we're not quite there with the combination. We're not ready to develop the CHOP versus something else as they do in cancer. But we are really doing add-on trial designs already with the combinations of riluzole, radicava, and now I call it Albrioza. You're calling it something else. I can't pronounce your drug name, so I'm going to call it mine.
I can't pronounce yours.
But at least we're at add-on therapy. So those steps are real for some patients.
Yeah, I want to add, that the IVA Derivan study was an add-on study. Everybody was on riluzole and then additional, and it's hard to do that in the US because of the insurance stuff. But there are ways, like if MT Pharma and Amylyx actually would provide the drug for these studies, you could have, if people wanted, they could have everybody on it and then check. And then the new one is really an add-on. That's one way to do it. But I do think that we need to start thinking about preclinical studies to make sure that these, even the 3 that we have, are at least at the minimum, not interfering with each other. Maybe they're also additive, and then anything new, making sure that we're not having some effects that we don't anticipate.
Yeah,
I mean,
I'll clarify about that. My thinking was really going more toward combinations of 2 experimental therapies.
Yeah.
I agree with everything that I said about.
I mean, one can make a rationale perhaps to use a terminology that filtered through the last 2 days, combining upstream and downstream interventions, right? So, I mean, the tofersen results are beautiful, but the treated patients are still progressing. And clearly we're going to need additional interventions once the death process, the morass that is the pathophysiology is underway, the momentum is there. We're going to need other interventions to make a difference. And so as one thinks about at least 2 drug therapies, there's a logic to looking up and downstream, I think. The problem is that the downstream options are so numerous that it's very, very hard to know how to prioritize it. But I do think what's interesting, and it's the same is true in FUS, too. In the FUS patients that were presented by Neil Schneider, the one patient, he clearly turned off FUS, but the patients still progressed. Yes, little stabilization, but we're going to need more in terms of downstream manipulation of the disease process.
So I'm going to
Probably-
introduce
probably
another
not early.
concept. Sorry.
Sorry, go ahead, Angela.
So there's something that I've been observing, and we don't capture it well yet in our clinical trials. And I think this is where some of the biology is really going to help us. And that's the concept of responders versus non-responders. Because when we look at the data, we're looking at the collective, right? And we're combining everyone's results on placebo and on active drug. But once you get into what people love to call pragmatic or real world evidence, you do see that there's potential for a spread of response. It's not well documented, it's not well captured, but I'd like to introduce the concept of responders and whether we, with our biology, can use some of that predicted response in our inclusion criteria, or whether we use it in our subgroup analyses to understand it better. Because I think some of the discrepancies in case reports that we see in the literature may actually be people looking at different parts of the elephant. You know, there are maybe patients, we clearly saw it with tofersen. We had enough patients that we could see a difference, that certain patients clearly had a more impressive response than others. How we capture that, I think the scientists have to help us on that.
Yeah.
Well, I think that's part of the challenge of an extremely heterogeneous disease is how do we take a single measure or a handful of measures and try to apply them broadly to any given drug where you may have different responses across the population. And I think we're only beginning to learn really what's going to go on in that area as we have more and more options that we can look at, both in terms of therapeutics as well as biomarkers.
Yeah, it's interesting because I think that verges on the discussion of stratification. So one can look, as you said, Bruce, at the heterogeneity and stratify cases a priori by virtue of, for obviously genetics, for example, or style of clinical presentation or rate of progression in the first 6 months. Or alternatively, one can do what Angela's just described and do a retrospective stratification and ask who responded and who didn't and what separates them. But I think either way, the attention to the heterogeneity and defining the distinctions, either at baseline or in response to therapy, is one of our big assignments.
Yeah, I think as we're getting more data like PROACT and natural history data, we have companies like Origin and other groups that are starting to have some of those predictive models. And that might be one way to look at responders. But that requires very large data sets, which I know there's a big initiative to led really by the patient community to advocate for a large natural history study that would provide that type of data you need for those type of analytics.
Yeah,
Bob, I want to go back to the question you were asking about combinatorial therapies, and I want to ask one that's a little more specific, which is we may be in a situation where we have both ASO and gene therapies with the same gene targets. SOD1 is one that comes to mind, but you know, it could be true for any of the gene targets that we're targeting. And I wonder what your thoughts are about combining modalities to target the same upstream event and how we'll approach that.
I think one of the things to say is we whether or not we need to do that depends on target engagement and the output. You know, so in our little N=2 trial of microRNA to suppress SOD1, in fact, you're the co-author. In the first case, we had very, very good suppression of SOD1 with the one reagent. So I don't think we'd want to add another reagent. Although quickly to go sideways on the comment, I think to have more readily useful reagents than something that requires, you know, cisternal or intrathecal infusion is obviously going to be very important. So the answer to the question is, I think it depends on what level of engagement we develop with each agent alone. So
What about, Bob, just to take James's thought one step forward, what about combining ASOs with monoclonals directed at the output? So
Yeah.
what I'm really thinking of is combining something like tofersen, which we know starts far enough upstream that it takes 3 months.
Yeah.
With a monoclonal that basically is targeting misfolded SOT1, or the same story in C9 or in FUS, where you have a dual action, removing what's circulating at the same time you're reducing the gene product.
Yeah,
Something
no,
to think
I agree with that. And in fact, I mean, there are ASOs being developed now that target more than one thing on the first infusion, some of which could be upstream and some could be the downstream targets, as you're saying. So, yeah. So, Merit, you use the word natural history, and I'd like to pick up on that if I may. I think that there are 2 or 3 issues that brings up, which were, I think, addressed off and on during this meeting. The first question is, do we know enough about the natural history of ALS in different subtypes? That's number 1, just starting there, basically. And number 2, can you comment, or anyone else, on this terrible delay between the first symptoms and, of course, the establishment of a clear diagnosis, which has a big bearing on therapeutic window?
Yeah, I think, because of PROACT, we have, a lot of data and also recent data on people who are in trials. And that's a valuable data set that's used a lot. But it doesn't capture, the majority of people with ALS. And I do think there's a gap there in both the different genetic forms, to have really large data sets of people who carry the different genetic forms, as well as sporadic disease. And we don't have anything current. So there are a lot of small initiatives going on, but nothing that I think is the size that we would need if we actually wanted to use that data for predictive algorithms or for innovative kind of trial designs for rare subsets. So I do think there's a big need for that, and I hope it comes out of this NIH initiative as one of their top priorities.
Yeah,
I forgot your second question.
But just to pick up on what you just said, do you think we get enough biofluids, things like stem cells, PBMCs from each case? We have, for example, the wonderful work that Answer ALS did in terms of getting omics on the map with stem cells, but we don't really have a rich enough, I don't think, clinical database on each case by case. So
Right.
should we increase that whole effort?
Yeah, I do think we need that to increase, and you could see it in layers, right? There might be some things that you get on tens or hundreds of thousands of people, right? Maybe DNA and blood, and then subsets of people where you get the the PBMCs or the spinal taps or the more detailed fluid. It might be hard to do that on everybody, but I like James's input on the feasibility of doing that kind of work on large numbers of people.
Yeah, no, I think it's a great question. I mean, so when we collect blood, a lot of what we collect is plasma and serum, and those those can be collected quite readily at a center that does this. PBMCs are more complicated, so depends on the fate of them. So PBMCs that are fated to become iPS cells can actually be sort of mistreated for a little while. Because they just need to be alive by the time we turn them into iPSCs. So a little benign neglect is okay, and that can happen in shipment in the mail. Where it gets more complicated, and I think everybody's still sort of working on this, is trying to be very productive with PBMCs that are fated for analysis of their sort of immune function or their gene expression profile, because that changes pretty quickly, actually. There are some new systems where we can use fixative buffers. There's one called Smart Buffer or Smart Tube. And so that might make it practical. We're sort of working with that here, but it is a much bigger effort to collect cells that are, cells for immune analysis. I would also say that, we have the NEALS biorepository. There's the Target biorepository that's coming. The CDC has a biorepository. Answer ALS has a very uniform one. There's an opportunity in trials, though, I think that you're hitting on, Bob, that is really, really wonderful, which is that, people are motivated to be there. They have to be there over time. We don't experience nearly the kind of dropout in trials as we do in observational studies. So there's a real opportunity in including what we can in trial.
Yeah, I think that's right.
Yeah. I'd like to loop back to your second question, which is the issue of time to diagnosis.
Yeah.
And I think there are a few things that would make a really big difference. The first that I think we can work on today is just better education of the general medical community about ALS. Having them understand what the symptoms are, having them understand what to go looking for, having primary care physicians not be afraid to order an EMG, as mine was not afraid to do, rather than take the patient, send them to a neurologist orders an EMG. By the time you get the readout from that, you've blown 6 months of the diagnostic process. The other part is better biomarkers, particularly for pre-symptomatic patients. In the ideal world, someday we would have some sort of a very definitive biomarker test, blood test, CSF, something like that, where you could take it, you could measure, and you could say, aha, we have someone who is clinically significant heading into symptomatic ALS.
Yeah.
So
Yeah.
I think there's one other piece here, and Bruce, I'm so glad you brought it up. The other piece is, why does the general neurologist in the community need to do 100,000 tests before they send them to the clinic? 100,000 is a little bit of an exaggeration, but I think maybe that's what it costs in the US, but what we see and we have terrible time to diagnose these in parts of Canada. What we see is the neurologist who gets the patient first feels obligated to run through absolutely every test before they refer them on to the centers. And what we'd really like to do is change the thinking in the community. If you think it's ALS, you send it. You don't wait to do your own round of MRIs and EMGs and repeating EMGs. You send them to where they can get service, get a diagnosis and treatment right off the bat.
A great point. How do you popularize that with the broader community?
Yeah.
So I would point out the ALS Association, and Suma Babu, who's at our center, was really involved in this, did come up with a tool called the Think ALS tool, which is available online. And it really is a thoughtful approach to sort of, limb onset, bulbar onset, what things might support it and what things would work against it. I think we need to figure out how to use a tool like that, get people educated about it, know that it's there, because a lot of PCPs will see this once in their career. Neurologists don't see it very, very frequently, and I think it's helpful to have some decision support tool, even if it's just a card that says, here's what you think about. The other thing is, as our treatment options change, the urgency to make the diagnosis changes, but hopefully, along with that, the willingness to entertain the diagnosis will change.
Yeah.
So we found one major caveat to that, James, and that is neurologists in the community who misunderstand the terminology around LS Scorial. So
Yeah.
I'm hoping that Gold Coast penetrates the community, because there are neurologists that make the diagnosis when the patients first come in, but they do not send them on to a clinic until they reach the category of definitive, which is way too late. So there's some rethink. There's one group that's been pushing for the same concept as we have in stroke, where it's an emergency, send them on. But anyway, that's my thought. It is really important, because those patients never even get the opportunity to participate in clinical trials. They're sent too late.
Yeah, no, that's so true. And you know what, as I think about this, your points are so well taken, Angela. When I think about, particularly with bulbar patients, where one finds nothing in the limbs, here's the dysarthria, an ENT exam is negative, as is an MRI, and one knows that is going to evolve into LS Scorial positive motor neuron disease. And it's just, some of those take weeks to go to that next step. And it's just wrong not to get them into some kind of study program. That's so much the case. So there are some interesting comments in chat. I have several more questions of my own I'd love to put to the panel, but I think we should look at the chat. Professor Merlini makes the point that apropos of our discussion of how long a study should be, in transthyretin amyloidosis, none of the drugs approved and now available in the clinic showed superiority to placebo at 6 months, even using biomarkers. But it took 12 to 18 months to establish efficacy. So just put that out there as more food for thought in this realm. Becky Morey says, since EAP through Act for ALS will have a research component, will it be more restrictive regarding what you can take versus a traditional EAP? Any comment on that?
It's possible that so there, we use the EAP term as a fairly monolithic term. But there are slightly different designs within EAP. And I think over time, as we get more sophisticated about this, we'll probably think of these, codify these differences a little bit. Some EAPs are happening while the FDA deliberates whether there should be approval. We saw this with Amylyx. And there really wasn't much of a question being asked. It was really about getting access to people in the community who, to a drug that looked good and was being considered for approval. It really was an access thing. But we're also using expanded access to accompany and expand the safety data that we can get and expand the kind of people with ALS who can have access to experimental therapies earlier in development. And we may be asking different questions. We may know less about safety, and we may have to be a little more restrictive about what other medications people can be on at the same time.
Thank you. Another question, Camille Mekowaris, are there some drugs that just barely failed at phase 3, i.e. they had a apparent positive clinical effect, but their p-values were just above p equals 0.05? And is there a way to resuscitate or reconsider those?
I can't actually think of one. It's a good question. I think because in this field, because of the big unmet need, I think if something is trending or close, there's usually a follow-up on it. It isn't abandoned, but I could be wrong about not remembering one.
There was one. There was one, and it was a long time ago. Only a couple of us were born. And it was with Sanofi. It was called Zaliprodin. And there were a lot of issues around that program because Riluzole was only approved in certain countries. So in fact, there were 2 trials. And this is where what we've learned about biomarkers could really change the game, right? If they were near misses, if we had an immune biomarker or neurofilaments or something else, could we have actually met that target of statistical significance? And so the other thing I want to fit in, I'm slipping in my own comment here, Bob, because I'm thinking back to Bruce's comment about the fact that we still have to do placebo-controlled trials, and that's a regulatory decision, right? And there are reasons, not using historical controls. And now there is a group of, there's a technology, well, it's more than a technology. There are companies that are AI companies that are mostly focused on dementia because it's a much bigger prize at the end of the rainbow. But they are developing sort of virtual patients. And the idea with these companies, and they're early, they're starting to talk to the FDA. They were part of a panel a few weeks ago. But the concept is every patient gets treated and you develop your holograph or your meme or whatever,
Digital twin.
patient control using AI so that every physical patient in the trial gets the drug, and you create a placebo arm virtually. Now, what that would do is 2 things. One, it means the patients would not have to be on placebo, at least as soon as you could start using these substitutes. 2, we could then get to slightly longer trials and be able to reintroduce some of our other concepts statistically, because we're not leaving anyone out on placebo for an ordered period of time. I don't know how long this will take, but I find it a fascinating development to be able to create these model placebo arms that would allow us really to treat everybody, but allow us to then therefore extend the trials a little bit longer and do some more creative things.
That's fascinating. So Sava asked this question: are we near the stage where we could rank the main pathological mechanisms in ALS by relevance, i.e. lipid toxicity, above gene expression, above protein misfolding, et cetera, et cetera? I'd love to hear what you folks think.
That's a tricky one. So it does feel like we've got a lot of evidence for, for a handful of, maybe a little more than a handful of pathways being common across different models of ALS and in tissue, in human tissue. And I guess what I would say is that one of the things that we're trying to do, and you hear us wrestling with it, I think, is put these in order of sort of more upstream and more downstream. And sometimes that's easy to do. I mean, obviously we talked about tofersen targeting upstream, but some of the rest of it is a little bit harder to do. And so, and the other thing that's funny is that much of it does not confer a cell specificity, which is still a little confounding. My wife is a psychiatrist, and I get a little worried when she puts her slide up about what's going wrong in schizophrenia, and I put my slide up about what's going wrong in ALS, and we kind of agree on a lot of it. That's a little worrisome. It means there's probably something more. Anyway, I don't know if that's an answer at all. It's just amusing.
Yeah.
I was gonna say, I don't think that we can now. I mean, and I think it would be a mistake to try to do that, because we'd probably be wrong. It's not just that how to order them at one time. It's that there might be different steps of different parts of the illness that are more important, or different groups of people. And we need to get to that point, but I don't think we're there yet to be able to, rank order biologies.
Yeah. I mean, the complexity, in fact, Erica showed that extraordinary image of a motor neuron with an axonal process, which is 10,000 times longer than the width of the motor neuron. And the complexity of the death process is just dazzling. I even flinch when I hear people say ALS is a dying back disease. I'm not sure that's true at all, because when the soma gets sick, the structures that maintain the axon are going to necessarily fail. And so whether or not we can impose that view on ALS and make it equivalent to, say, a motor neuropathy, I think is crazy. But Sava, this is a crucially important question, and it's one that we need to continue to pursue. I would say one other thing, which is it was very good to see the lipid toxicity and oxidation story today. It's quite interesting that when one looks at recent transcriptomic studies, the disturbances of cholesterol metabolism are actually quite common in ALS. We've gone through the decade of the brain, the decade of the genome, which essentially are decades of genes and proteins. And I think we're still just beginning to come to terms with lipid derangements. So, I mean, there's at least a whole nother chapter to be written there. I really believe that. Okay, let me just quickly check other questions here. Let me turn to a slightly different issue that I think we as clinicians perhaps could talk more about. And that is, do we do enough to address quality of life issues in ALS? We're so concerned, and wonderfully so, in a meeting like this, in extraordinary molecular biology and therapeutics, trial design and drugs. But what about quality of life? And maybe, if I may, can I turn to you, Bruce, and get your view first? Do you think we have done enough to address quality of life issues?
As I think about my situation, part of my working with quality of life comes from the clinic. Part of it comes from resources outside of the clinic, like Compassionate Care ALS. and part of it comes from my broader community. So it's all of that mixed together. In my case, what I've found is if I can figure out how to articulate the question, often the team at clinic can then start guiding me towards the right resources. But that takes first and foremost the presence of mind to try to figure out how to articulate the question that you're dealing with. So, and I can give you a great example. I had to start using convent catheters at night about 4 or 5 months ago. The startup process was, to put it mildly, painful because I needed to talk to a whole lot of different people about all the different issues around that. And it got to the point that I finally realized, wait a second, there's some room for education here. And I wrote up a 5-page long document about my maintaining nighttime urinary independence over the last 3 years. So, and that now circulates with the team at MGH, but it's again an area where I worked as a patient to try to help give feedback for where quality of life issues could be better addressed.
Yeah. I mean, I worry about this in one of the groups, ALS Finding a Cure, which has been so beneficial to the field. We spoke for a while about making a series of videos on different components of the disease and trying to deconstruct what some of these are. And, you know, as I think back, I think that'd be a wonderful thing to do. I know, Merit, you have, of course, a wonderfully and I'm sure Angela does as well, multidisciplinary clinic, which goes a long way to addressing these things bit by bit. But the question is, do we really do what we need to do?
Yeah, and I do think there are some foundations that are tackling this. I see in the chat, I am ALS. There's also another one, I'm blanking on their name, who's making videos with patients to cover each of these kind of topics.
Are you thinking about your ALS guide?
Yes,
because they've got a very good video series.
Yes, exactly. Yeah. So I think the clinics are doing their best, but there are many people, as we know, who don't live near a clinic. So we need education material that's easier to access. I'd say in trials, we struggle a little bit with how to measure quality of life. I mean, the tools we have now, and really Zach Simmons is kind of the leader in this from Penn State, they actually show that quality of life doesn't change with the illness. It's what's important to people that changes. But that's not, because sometimes people ask us, did the drug improve our quality of life? And we're not able to quantify that or measure that. So there's some work that needs to be done there as well.
And if I were to chime in a bit, Bob, I think we should never forget the importance of symptomatic therapies and really sharing that, because those are quality of life issues that really make a difference. Whether it's approach to salivation, whether it's approach to the GI issues that nobody writes about, that's a big problem for the patients. Whether it's, we forget how important the development of Nuedexta was. Yeah. And it's asymptomatic. It's got some evidence for being disease modifying, but these kind of interventions are not as well known as they should be, because they're not the big splashy clinical trials that are what everyone focuses on. And most of the interventions in that symptomatic are available to smaller communities and docs but the information hasn't spread far enough. It's just a bit crazy if someone is from like even another province and no one's talked to them about Botox for hypersalivation. It makes no sense. Yeah. So yeah, I think there's that layer too.
And Bob, I think we'd be remiss to not talk about some of the reimbursement for clinics. I mean, we're most everybody is running a multidisciplinary clinic and funding that through philanthropy and grants and wherever and just the goodness of the hearts of the people who are working there and not getting paid for their ALS work. Whatever it is, different institutions are different. There's not a sort of clearly thought out plan to support the multidisciplinary clinics that we think are really valuable. And so that I think is an opportunity for improvement. And I'll say one other thing, which is that it really does take a village. All of the organizations that are helping people with ALS have a real role. I was very struck early in my career when I went to see a patient at home who I'd been caring for in clinic. And her family was leaving town and she needed a caregiver and she needed just an insurance form signed, but had to be a face-to-face, and we weren't doing telemedicine very early. But I'd followed her for a year or so, and we had lots of things set up at home. And she had every piece of equipment, all the durable medical equipment. There was a commode, and we had BiPAP and suction and Hoyer lifts. So bad. And I got to her house, and everything was in the living room, and she was in her bedroom.
Wow.
And the doorway was too small. You couldn't. So none of the equipment that we had diligently sort of ordered and she had been so thankful for, but I think embarrassed to tell us that she didn't know it wasn't working at home, or shy to tell us that, to disappoint us.
Yeah.
Boy, I saw a real disconnect. And,
I
So
would
I-
mention, yeah. No, yeah. And two sort of side comments. One is how often do we, as if I guess I could say that, the primary ALS docs actually go into the homes and see patients. I mean, long gone are the days when that was routine. Many of us have done it from time to time, but what a difference it makes for so many reasons. The second, yeah.
Well,
go ahead.
and I'll just say we actually on the basis of that we wrote some grants, and Merit and I worked on this and have funding from a foundation to help with the house call program, that we can at least reach people around or surrounding our clinic. But it would be nice if it was more widespread, the real need.
Yeah, and I think that's one of the great things about agencies like Compassionate Care ALS, whose sole purpose is to do exactly that, to get into the home and see people. The other side of point about quality of life, and which is, I think, so true, is the importance of quality of life for the caregiver. I mean, if we all know so well the scenario in which a caregiver gets sick, unwell, has some disability suddenly arise, and how devastating that can be for the overall care plan for the individual with motor neuron disease.
Yeah, thank you for acknowledging Peter Werner's comment in the chat
Yes.
with that remark. It's incredibly important, and there are days that I will freely offer that I think the quality of life for my wife, who is my primary caregiver, is impacted even more than my quality of life with all of the physical and emotional symptoms that come with ALS. And it can be a real challenge for me to see her struggling. And then there are days I occasionally think, oh my gosh, she's not feeling well. What would happen if this got a lot worse? So it's a huge, huge issue. And I think caregivers don't get enough attention. There's so much attention focused on the patient because the patients need it, that oftentimes the caregivers are just overlooked. And I can tell you that anytime I'm meeting with people or I'm in an appointment and somebody simply turns to my wife and says, "And how are you doing?" it makes a big difference. Just that one simple question and acknowledgement.
Yeah. Yes, and thanks to Peter Werner for this comment. A couple other things. There's another question in the chat. Are there studies showing possible diseases or health issues that the patient with sporadic or familial ALS often present with at the time of disease?
I know that there's a poster that Nazma Tassi worked on with, I think it was with people at MT Pharma, actually, if I remember right, where they looked at the occurrence of, it was in an insurance billing database. So, and they looked at sort of people who were diagnosed with ALS and then the preceding diagnoses. And which I thought was interesting. And there were diagnoses that we might think are disease mimics, kind of mixed in there, or not disease mimics, that's a better way to say it. Things that symptoms were attributed to. Reflux, for example, rather than dysphagia. Is just an example that comes to mind.
Yeah.
Ridiculous.
I don't have a real, oh, sorry, James.
The last thing I was going to just say is, it was what would be interesting is if there were other pathologies that were riders along with ALS or diseases that preceded. I don't know that that was so clear or easy to disentangle.
Yeah, no, it's a good point. I see ALS as one of those conditions where people, they just, as the expression goes, they just have ALS. They're perfectly healthy, running their lives, and this hits them, and it contributes to denial and disbelief at the beginning of the disease, because you can't imagine that this little problem with your hand is not just a little problem with your hand. The other thing that we didn't talk about in quality of life, Bob, and I would be remiss if I didn't bring it up, is the spiritual health and the motivational health of the patient and the family. We've had a spiritual counselor as part of our multidisciplinary team since the 90s, and each brings their own skill set to the team because it turns over years. And on one occasion, they did a survey to try to determine what is the most important features in terms of quality of life. And there were a couple of things that really stood out in that survey. One was hopefulness. If they maintain a sense of hopefulness, they do better. No matter what the clinical presentation, if there's a sense of hopefulness, that's a huge driver for quality of life for the patient and the family. The other thing that really stood out was purpose. If they were still somehow valued in their family and their community as giving something important, whether it was going to, remember I'm from Canada, going to their grandson's hockey game, being a fixture there, week in, week out, little league, midget, whatever, but a sense of purpose, that they're needed in their environment. And that environment doesn't have to be family. It can be social. It can be their church. It can be their work. I had a patient who was still going in as an accountant until 3 weeks before he died. And he had a pagan event, and he went to work, and they had an office, and it contributed substantially to his quality of life. More recently, two things have happened. Another group on the team have started looking at the quality of life of the caregivers and to see what triggers. They've done one poster. I think they have another one coming. And a lot of that is the support that what the caregiver can maintain of their life before their loved one developed ALS. And caregivers in this instance can be kids, it can be parents, or it can be spouses. And then the final thing I'd say is our current spiritual counselor actually has been working with someone from Boston. A couple of academics from Boston has developed a mindfulness program. And this mindfulness program is being offered to the patients, their caregivers, and the staff in the clinic. Because the staff have to be healthy to do this work. And I'm really impressed by all three sets of insights that have come out and how they influence. It actually influences people's willingness to go into a clinical trial. If they're hopeful, if they feel there's potential, or if they feel that they're contributing, they have a purpose, they're far more likely, apparently, to go into a clinical trial and to be part of what we need to do to figure this out. So just some thoughts.
Those are really, really great thoughts. And I think, as you said, that issue of wellness and spirituality is probably the right word, one way or another, echoes, as you say, through the full clinical entity, not just patients and caregivers. I mean, I can't tell you how many times I've had patients say to me, how do you do it? I'm sure everyone in this call has had that question. How do you do it year in and year out? 30 years go by and you're still seeing patients with this disease. And you know, for us, because of meetings just like this, there is always hope. You know, the flag is always unfurled and flying, and I think we're all very upbeat about where we're going, although it has not been as fast as any of us would like. But I think that's really, really important. And there's no doubt that I think patients, it's almost like transference, pick up the mindset of the staff administering or trying to administer the best care. There's a-
100%.
really,
I
really
was
good.
going to add to that, that we have a chaplain also that's available for our patients. But one of the things that they said to us right away, well, maybe after a couple of weeks, is we think we can help your team too. And we want to meet with your team. And they've been doing that. And it's, I think every clinic should do that. I think it's good for everybody.
Could I just also add to, I just wanted to say too that there are other organizations out there that I know of. There's one called ALS Family of Faith, and they're wonderful. And for people who want to bring, you know, to whether it's pray with someone or whatever, they kind of pair people with a mentor and kind of help them through that. But I wanted to just point out too that oftentimes people in Bruce, please correct me if I'm wrong here, speaking out of line, but people often have said to me that when other people greet them, it's almost like this sullen, like, I'm so sorry, sadness. And it actually, what you should probably do is the reverse. And that has been my practice, is when I meet people, or, you know, I try to emit hope and positivity rather than, you know, walk in and just be, oh God, I'm so sorry. Because not everyone, there's a lot of people who have ALS that are living with ALS.
Right.
And
And
that's
that's an
what
important
they'll say.
word.
Yeah.
Living with ALS is critically important, not battling it, not struggling it, not dying from it, but simply living with it. And the way I approach it is I try as much as I can to reach for the same degree of normalcy that I had 5 years ago before I was diagnosed. And that means just trying to live my life the way I best can. I know one of my work colleagues is sitting in on this meeting right now, and I think she would tell you that most work meetings, because I'm still working, I focus on work. ALS once in a while drifts into the conversation, but it's not the center of the conversation. Can I get it out of being the center of my life? No. My disability is far too large for that, but I still try to focus on, as much as I can, the normalcy of my life.
Yeah, it's important. Definitely.
So many issues arise here. We've heard the comment so many times about individuals who have got to the stage of ambulating in a wheelchair and having, at the start of a conversation or meeting someone new, having them make eye contact with a spouse. And somehow the fact that one is in a chair demeans or demotes or leads to terribly erroneous assumptions about disability. I mean, we see that a lot as well. And so many, you know, you could probably spend 3 hours, Bruce, telling us what some of these hurdles have been over these 5 years. I can't begin to imagine.
Some of it is disability as a mindset. I'm very careful, and I know this can be controversial in the community in terms of how I'm wording it. I do not think of myself as being disabled. I think of myself as having a disability. And what I try to do is use the resources at hand, whether my power wheelchair, whether voice recognition, other technologies, other tools for quality of life, to keep myself on the curve of, I have a disability, but I am not disabled. I can still do things. I can still be active in the community. And for me, that's a critical distinction.
Thank you. Thank you. We have just a few more minutes. There are a couple of questions in the chat again. And so one is from Shalina Lalji, who asks, would the panel please comment on which of the numerous and impressive presentations have the most promising data regarding recovery of function, which is not always, we always say, wouldn't it be wonderful if we could stabilize the disease? But she's asking, of course, about the dream hopefully come true of recovery. Who would like to comment on that?
I mean, I think.
Michelle and I talk about it a lot, so she probably doesn't want to hear my answer. So let's hear somebody else's. And I'll add in.
Well, yeah, you can go at the end. I mean, I think this absolutely is a goal, and you know, there, as we've now seen a few more recent successes in drugs that can slow ALS, I think we are now sort of allowing ourselves to think about these loftier goals of prevention and of recovery of function. It's, you know, it is a high bar, but I guess one thing I would say is that we know from polio, which is an infectious poliomyelitis, anyways, is an infectious form of motor neuron disease. And, you know, when that process halts, we do see recovery. We don't see complete recovery in everybody, but we see a natural recovery. And so, you know, the two things may not be so different, slowing of progression and recovery. It may be that if we can get to a place where we slow progression enough, we don't need to do the recovery. I mean, it would be wonderful to have ways to also regenerate. That's one way.
I think we go back to things we talked about earlier. The first thing is going to be getting these diagnoses, getting these patients on therapies much faster when the majority of their symptoms, they still have motor neurons there so that you can get some recovery of function. So I think once we really change that diagnostic pathway to 3 months instead of 18 months, we're going to have a much better chance. And the second thought I have is actually a combination therapy approach, because we already have drugs that can work or treatments like tofersen that work very early in the disease pathway, and simultaneously we're looking still at reldesemtiv, which is a troponin activator, which you could imagine a situation in which a patient with SOD1 ALS receives tofersen and then adds in reldesemtiv and actually does see a recovery of function, because they're no longer losing the motor neurons, and the remaining motor neurons are actually able to get this improved contraction through the second drug. So those two thoughts.
Yeah.
I'll just add, I agree completely. I think there's two ways to think about repair. One is drugs that actually help regrow axons or reconnect axons to the muscles. But you can see those, just like Angela said, would need to be in combination with things that are slowing down the illness. Then you can think of the body's natural ability, like in polio, to repair, that if you can get upstream enough and early enough that you block the biology, the surviving motor neurons might do the repair process for us.
Yeah, I would fully agree with that as well. So B asks the following: what do you think about alternative therapies such as acupuncture, ozone therapy? Do you see mechanisms such as these that might help ALS patients?
I guess I think that there's a lot of people who spend a lot of time and money on those, and I think just like clinical trials that they should be tried, because I think most of them don't work. I mean, there might be something out there that will work for people, but if we're not studying it rigorously, we'll never really learn. So that's I think they should have the same rigor of assessment as other treatments.
Yeah. Although the word alternative, I think, is so important. I mean, what we've seen so well epitomized in the last 48 hours is the paradigm, which is the paradigm about how we think about ALS. And I still worry a lot that we have built a lockbox around ourselves, and until we get out of it, we will not really be able to ask new questions. You know, one question we haven't talked about, we don't have time to now, is the huge set of elephants in the room, which is what is sporadic ALS? And that came up, of course, during these 2 days. But what we have not done a whole lot with, speaking of alternative, is thinking about interaction with the environment. You know, the brain microbiome, let alone the gut microbiome, and you know, the role of subtle and infective or toxic entities in disturbing the nervous system over a long period of time. There's just so much at play here. And one of the questions for us and this is for Jen as well, is as we think about ALS one and how to break out of our paradigm and think about really new innovative concepts in the disease, what does it take? Who do we want to get involved? Because this question about alternative becomes an important word as we think about paradigms. Anyway, that's just my own brief comment there.
So I do see another way of looking at some of the things that were listed. So acupuncture as a symptomatic therapy is actually quite successful for certain symptoms with ALS and can definitely improve quality of life. I live in a country where marijuana is now legal, but I've prescribed medical marijuana for many years, and it's been considered an alternative therapy, and yet it's extremely effective for some of the core disabling symptoms in ALS. So there are alternative therapies that I agree with Merit. They need to be studied properly. There's really no fact around them, but we shouldn't lump everything that we consider alternative in the same bucket. There is good evidence for acupuncture. There is good evidence for marijuana. I'm not yet in the field of prescribing psychedelics, which are coming on into mainstream care now. But there are things like this that I think we should be more comfortable using to improve quality of life and deal with some symptoms.
Thank you. I have certainly encountered individuals who spoke of much benefit from acupuncture. Although again, they're so anecdotal, I have to fight against, I have to be cynical about my own cynicism, because the problem is it's sometimes hard to accept that, but ideally these things should be studied. So yeah,
I mean,
go ahead, James.
Well, I think yes, we should study these things. I think we want to set up studies that are, most likely to give us answers we can act on. The chances that acupuncture cures ALS are, very astronomically low, and probably not going to be high on the priority of things to study. And I think what we run into is situations where we have to guide people about, about what they're hearing from an alternative medicine person, because the problem is that mixed in amongst there are people that are predatory, and that I think is challenging, but is a conversation that people with ALS should be willing to have with their providers, and their providers should be willing to have. But there are also a lot of things that we have an absence of knowledge about that could be helpful. I mean, acupuncture, the fact that we don't have large studies about acupuncture doesn't mean that somebody ought not to do it, only that we should have a conversation about it. And I think there are lots of examples of that in living with ALS, but just in living life. I mean,
Yeah. Well, I think we're nearing the end. There was one more question in the chat, which takes us back to diagnostic issues. This is a question from DuPont Therapeutics. What is or are the confirmatory tests that diagnose ALS? And I think this gets back to this question of how finally we acknowledge that we're painted into a corner. And I don't know who would like to take that, but it's James. How about you?
Yeah, maybe. I mean, I can. So, we often say ALS is a diagnosis of exclusion. That's partly true. So, I think we use the exam to look for signs that are consistent with ALS. We use the history to look for symptoms that are consistent with ALS. EMG. You can't do an EMG and in isolation look at that EMG and be 100% sure that someone has ALS, but there are EMGs that are consistent with ALS. LS4EL and other criteria sort of set out. Awaji also set out sort of some criteria for how to use that, both to rule in ALS and also to rule out potential mimic diseases. And then we want to rule out other potential mimics using imaging and lab tests. And with that, without without listing all of those in detail, that forms the basis of Escorial criteria, Wadji criteria, Gold Coach criteria. They're not that different in saying a progressive disease limited to the motor neurons or strength subsystem of the neurologic system, where you rule out things with imaging and rule out disease mimics with EMG and get an EMG that looks consistent with ALS. That's ALS. Now, neurofilament is clinical. This came up in the chat now too. Neurofilament is a clinical test that we can order now too. And I think, we have a long history of looking at it in a research setting. We have a short history of using it clinically. And I think we're going to have to figure out how we do that. But it's, again, neurofilament goes up in most neurologic disorders. Anything that injures motor neuron, anything that injures neurons will cause neurofilament to go up. So we have to be cautious about it. Some people with ALS do not have an elevated neurofilament. So it's yet another imperfect test, but maybe another test in the armamentarium. And there are others that are hopefully come.
Yeah, we sorely lack something like the troponin leak of a myocardial origin that you see, for example, with an acute heart attack. We'll get there. We'll get there.
Although we may have the troponin leak that you get with ALS. I mean, troponin T actually. It's funny you should mention troponin leaks.
Is that evolving, James?
Troponin T looks like it. So Mary can maybe talk a little bit more about the trial, the Orion trial that sort of originally looked at this. But I'll also tell you anecdotally, our center changed from troponin I to troponin T, and troponin T is just in patients in the ED. Troponin T is a little elevated.
Yeah, interesting. Wonderful. Well, I think this has been a wide-ranging but very appropriate and very, I think, very wonderful discussion. So I look to Jen and to others to conclude the meeting. But thanks to all members of the panel.
Thank you.
Thank you.
Yeah.